Glad to chair this session. And can I have the first slide, please? The session 3 is on biostimulant, post-regulatory perspective and prospects. It is becoming a very hot topic now because across the country, people are wondering what the regulatory part will influence on their business prospects. So the regulatory regulation is always a dynamic process. Ups and downs will be there. All of a sudden, all these products, when it is introduced into the market way back some 30, 35 years before, and all of a sudden when the regulation comes, industry is feeling not very good about the regulatory part because of some of the aspects like pesticide residual level, permissible level, as well as the toxicity study requirement, all these things. So now, majority of the biostimulant, some of the parts, Mr. Korakiwala has already covered. And now in this session, it is a very dynamic session with that focuses on plant residual management, stimulant and plant residual management, and the soil health management, then biostimulant for the efficient nutrient utilization, enzymes, vitamins, extracts, and finally, someone is going to talk about the regulatory aspects, what we are facing right now. So we have the distinguished speakers, I am pleased to introduce Dr. K. Annapurna. She is going to talk about the microbes as plant stimulants for the crop stubble management. A recipient of Lifetime Achievement Award on Microbial Agricultural Sciences, Dr. Annapurna needs no introduction. A product of Indian Council of Agriculture Research, Dr. Annapurna spent about 35 years on microbes of agriculture importance. She has spearheaded the Pusa decomposer technology among the farmers of Punjab and Haryana farmers to avoid paddy stubble burning and air pollution. A recipient of IARI Best Teaching Award, Dr. Annapurna going to talk about microbes as plant biostimulant and for crop stubble management, who also developed the carrier and liquid-based biofertilizers when she was heading the IARI microbiology division. And welcome, madam. And the next speaker is Dr. T.G. Prasad, the scientific advisor to P.J. Margo Private Limited, Bangalore. The Indian Society of Plant Physiology, a reputed scientific forum in India, bestowed him Life Achievement Award for his research and teaching contribution on plant physiology. BIPA heartily welcomes Dr. T.G. Prasad, scientific advisor to P.J. Margo Private Limited, as one of the speakers of the session. Dr. Prasad will be talking on biostimulant for nutrient use efficiency optimization. Dr. Prasad is also served UAS Bangalore as a retired professor, emeritus professor and head, Department of Crop Physiology. He guided over 20 doctoral students with 150 research publications of national and international repute. We are going to hear about his 40 years of his research experience and the commercial expertise on biostimulants. Heartily welcome, sir. And the third speaker is Professor Naveen Kumar Arora, the Dean, the School of Earth and Environmental Sciences, Baba Sahib Bhimrao Ambedkar University, Lucknow, UP. And he is also an editor and series editor of Springer Nature and one of the reputed journal. We are proud that he is the editor and series editor for Springer. And he is also a President, Society for Conservation of Wildlife, Secretary General, Society for Environmental Sustainability, an expert on microbial research with collaborative research in India and elsewhere. Very recently, he has come back, yesterday he has come back from Uzbekistan and spent some few hours with the family and came back to attend this session. Hearty welcome, sir. And the fourth speaker is Mr. R. Manoj Kumar, the founder and managing partner of Natura Crop Care and the recipient of Global Best Innovation Award. And incidentally, he is the student of Dr. Professor M.S. Rao, who is the advisor to BIPA. And he was the principal scientist of IIHR Bangalore and a product of IIHR. And sometimes expertise matters, sometimes innovation and the bold decisions are matters. He is the young budding entrepreneur who has decided to take his career on biologicals. Welcome, Mr. Manoj Kumar. And the last but least, Mr. Sahil Malik, founder of Kisan Agrovet and Joint Secretary Agri-Input Formulations and the Secretary National Biostimulant Industry Forum. I have to mention about the National Biostimulant Industry Forum, and BIPA is also a part of it because government is not able to receive communications from so many channels. So they have requested to form us a single platform, whereas some kind of NBF can be interacted with the government directly. So all the industrial forum came forward and created a platform called NBF, and he is the secretary and wide experience in interacting with all the government officials in Delhi on biostimulant. He will be sharing his experiences on the biostimulant regulations and what is next. And finally, He will also talk about the requirement of the data, especially I have also gone through when we are introducing zinc, boron, manganese, magnesium, urea, DAP, MOP, there are no tax package, there are no such kind of regulations. When traditionally when we are using this, regulation is required, we also agree, BIPA also is debating that, and microbes are one of the important factors that are not been included so far. BIPA is also working on it, and regulation is definitely required to streamline the products, but at what level? The benchmark should be to be met by the industry. It should be suitable to the industry. So this is what we are working for. So with this session, I request Dr. Annapurna to start her talk. Thank you very much.
Good evening, ladies and gentlemen. At the outset, I would like to thank BIPA and all the organizers for inviting me here to be a part of this program, and also giving me the opportunity to speak a few of my thoughts on how microbes can be used also as biostimulants. I will try to restrict my my talk or my viewpoints only on the aspect of microbes as biostimulants, because I do see that the rest of the speakers will be covering the other topics. So, and as part of the initial committees which were in ICR, which was formed for coming out with some kind of guidelines on biostimulants, I was surprised to see that microbes were completely omitted out. That's a huge mistake or blunder, I would say, and now we are trying to rectify that ICR level so that microbes can also be included as biostimulants under the guidelines. But the Gazette notification which has come out, there is no mention of microbes in that. So if as a plant physiologist, who is the next speaker, I would say that when they talk about biostimulants, they would always think about the impact of any molecule on the plant physiology, which means it can be biochemical, morphological, or molecular levels, at molecular levels, if I am correct. So this is how the stimulant word is being understood, and if with that context, if I take microbe as a biostimulant, then I would like to segregate the component of microbes as nutrient providers or any other nutrient providers basically, or also other stress ameliorators. But if I just speak on microbes as stimulants, then let's see what these microbes can do in terms of plant physiology and biochemical aspects. So here I've just now listed out some of the microbes, and I won't go into those details, but these are very well known as biofertilizers, whether it is Astobacter, Rhizospilum, etc. But the categories of biostimulants are already here, like microbes, which I would like to include, and biochemicals, protein hydrolysates, amino acids, vitamins, cell-free microbial extracts, antioxidants and anti-transpirants, and also the natural organics. This audience is already very well knowing about it. And the next I would like to say, when we are talking about microbes as stimulants, these are the 3 points I would like you to concentrate: where, who, and how. Where are these microbes present? Who are these microbes? And exactly how are these microbes being used as stimulant? What are the mechanisms by which these microbes are stimulating the plant physiological conditions for the improvement and for the better plant growth? So here I have just given on the right side panel, if you see, there is a plant, and you can see all kinds of, whether it is leaf pathogens, root, carbon exudates, etc., etc. But this one will exactly give you. And like Dr. Reddy has already mentioned, now we are talking in terms of microbial communities. So this slide will just give you the whole context of a microbiome. The top is air microbiome, ubiquitous. Microbes are all ubiquitous, so they are present everywhere. Air also has microbial communities. Phylospheric microbiome also has all the leaf plane, leaf stomatal openings, everything is also all having microbial communities. Then the rhizospheric microbiome, which is the soil adhering to the root, that also has microbial communities, as well as the soil microbiome and the endophytes, whether they are leaf endophytes or root endophytes. This is, I'm talking about the location, the where part of the microbiomes where they can be termed as biostimulant, but this is the location. The next comes the who. You can see a plethora of microorganisms which can be termed as biostimulants. But then again, biostimulants specifically should be stimulating the physiological processes of the plant, and there are different mechanisms which I'll shortly tell you. But here I've just given you an idea of all the different microorganisms, whether they are fungal, whether they are Actinomycetes, Rhizobium, or any other bacteria, whether they are red, blue, white, yellow. These are all the phenotypic and the functionally diverse microbial communities which can be termed as biostimulants, provided they actually have a mechanism of improving the plant physiological status. So how they are improving the plant physiology? The first one on the left side panel, if you focus on, this is through root and shoot growth, either through the indole acetic acid productions or the ACC deaminase enzyme. Or on the right side, if you look at the point 5, this is through plant defense, either through the ISR or the SAR mechanisms. So these 2 points are, I personally would say, microbes can be used as biostimulants because they are effectively impacting the plant physiology in terms of either responding to the plant's defense mechanisms or for biotic or abiotic stress manipulations, or as plant growth regulators in terms of their root growth, root elongation, etc. You all must have known about the abscisic acid and the salicylic acids. These two are plant regulatory hormones, very important hormones, and they are the ones which actually trigger the plant physiological system. And these two hormones are also produced by many of these microbes. There is no way we can distinguish the SA or the ABA which is being produced is microbial in origin or plant in origin. We really do not know how to distinguish that. But if these two molecules, these are signaling molecules, plant regulatory hormones, and extensively they have a role to play in all the plant growth conditions, like whether they are germination, bud formation, growth, or defense mechanisms in terms of abiotic or biotic stress conditions. Now, the beauty of that is, because these molecules are common to both the microbes as well as the plant, their biosynthetic pathways are also the same. So when we are talking about SA, that is salicylic acid, it is majorly a plant hormone which is stimulating the plant defense mechanisms for combating the disease, or for combating the any microbe which will be attacking it and causing a disease. So it is known for years together now, SA is known for defense mechanisms, eliciting defense mechanisms in the plants. Now in recent times, if you read the papers, SA is also being touted as one of those signaling molecules which helps the plant growth regulation in terms of root growth, root elongation, meristematic cell multiplication also. Now ABA is abscisic acid, which again is produced by the plant as well as by the microbes. So it has a useful or regulatory mechanism in all aspects of plant growth, whether it is simply the physiology of it or the morphology of it or the defense mechanism. I will leave the abiotic stress management to Dr. Arora, but if I just take overall the microbes as stimulants, I would like to stress upon these. When you are labeling a microbe as a stimulant, biostimulant, then you should know the mechanism behind it. Otherwise, a microbe can be a biocontrol agent, or a biofertilizer also, because it has ample role. And that does not mean that these are exclusive to each other. If a microbe is producing SA and is improving in triggering the defense mechanisms, stimulating the gene expression in the plant, then it does not mean that it is devoid of P solubilization trait or nitrogen fixing trait. It also has. Many times these are multi-trait microbes, so that we should not forget. But then labeling of that microbe as a biofertilizer or as a biostimulant is something that we have to do. So when we understand the mechanism, then only we can categorize them as a biostimulant or as a biofertilizer or as a biopesticide. So this is something which I would like to emphasize to the audience here, because like I said, without going into details of the rest of the highlights which I have given, whether it is nutrition or abiotic stresses or even the biocontrol aspect of it, I will only say that biostimulant, any molecule which is a biostimulant, whether it is a cell-free extract or a microbe, needs to impact the plant physiology. And as a physiologist here, he would, I'm sure, will be emphasizing on that. And this is what we missed out in the initial round of discussions at ICR, and by which that was one of the reasons, because the committee had only physiologists, did not have a microbiologist also, which actually should have included microbes also as one of the components in the earlier guidelines. The Gazette notification has also omitted out microbes. And here I will, in passing, will also talk about a little bit on the decomposition aspect or the crop residue management of the, by the microbes. Again, we do not have any guidelines formulated for this particular aspect. I think we should come out with, like you have biofertilizers guidelines, ISI guidelines, or FCO guidelines, we should also come out with some guidelines under FCO for biodecomposers also. Because this again is a different category altogether, which will be producing those lignocellulosic enzymes, which will be acting on the residue material, plant residue, and releases it. Whether the after impact is soil health conditions getting improved, soil nutrient status is improving, or soil carbon is improving, that is another matter. But the decomposition part is something which is taken care by the microbes. So I would suggest that if this August gathering and the companies are coming together with some kind of guideline, we must bring out guidelines for the decomposition part, so that FCO can also, the decomposers can also come under FCO, and it will be much more helpful for all the industry people. This is what I would like to say in my short presentation. I hope I leave something for you people to think about. And with that, the last slide of mine would be, like rest of us, for any biological product to work, it has a lot of hurdles to pass through, which means I talk about the constraints, the survival about it, the establishment, the growth or the multiplication, and the function. Importantly, the function as far as biostimulant is concerned. So if you are talking about microbes as a whole, you need to understand all the 4 restraints and constraints that I have just now listed. If they do not grow, they will not be able to produce those molecules which are necessary for triggering the plant physiological processes. And remember, in microbiology, we are taught about quorum sensing. Any microbe, and I come back to what Dr. Reddy said and what we are now talking about microbe. If a cell is just about 100 cells or even 1,000 cells or even 10,000 cells, it may not still be producing that particular molecule in sufficient concentration for it to be impactful, which means the cell or the microbe needs to be more than 1 million or so for the concentration of that molecule to be in such a dose that it will be impactful. So quorum sensing is one natural phenomena wherein we talk about microbial community, whether it is biopesticide or even biofertilizer or any molecule which produces an extracellular compound, that concentration will be enough or impactful only if the threshold limit of that cell number reaches one particular count. Below that, it will not be. It has the capacity, it has the potential, but it still will not be effective. So this is another important factor which, as microbiologists and industry people, we must always remember. The organism is there, but the number of that organism is not enough for it to be impactful. So with that, I'd like to take complete here. And thanks once again to all of you for giving me this opportunity.
Biostimulants and nutrient use efficiency. Thank you. So I first of all thank the organizers, the BIPA, on behalf of PJ Margot for inviting us to give a very brief account of what is the use of biostimulants in nutrient use efficiency. I'll go to some basic aspects and then tell how biostimulants are very much useful even in nutrient use efficiency, in addition to their role in a number of physiological biochemical processes which makes them to be called as biostimulants. So this is one important slide which clearly shows that in the last 6 years, there is still increase in the organic fertilizer use. You can see either urea or DAP or potassium fertilizer or the compost NPK together, still it is increasing at the rate of 10% or more. But you also know that every one of us know that most of this fertilizer we add in organic fertilizer is being not used up. To give you one simple example, as shown in this figure, in a paddy wetland ecosystem, you can see there is nearly 50% is only the nitrogen use efficiency. Of course, it is more what I am telling. It is less than that. As far as phosphorus is concerned, it's only 5 to 10%. And potassium is concerned, it is 40%, even less than 40%, clearly indicating that whatever we are applying is still there in the soil. And a small portion is being leaching out, and many amount is being still fixed in the soil and is not available for the plant. This is where the biostimulants also works. The biostimulants, in many cases, they attract these nutrients and make it available for the plant. It reduces leaching. It increases nutrient efficiency by a number of ways. Let us see one by one. So I don't want to go into these things, as Madam clearly told that to call it as a biostimulant, it should stimulate physiological processes in plant, in addition to a number of other processes also being stimulated. All of them can be called as physiological and biochemical processes at a gene expression level as well as at molecular and individual cell level. Biostimulants does a lot of things. It has a number of roles to play in plant growth and development physiologically. Starting from germination, seedling establishment, the development of the canopy, development of branches or tenure, and also giving stress tolerance, abiotic stress tolerance. In addition to these things, the two important things as far as this lecture is concerned, it also reduces nutrient leaching and improves nutrient uptake as well as nutrient use efficiency. So it helps in nutrient availability, nutrient uptake, as well as nutrient use efficiency. I am concentrating only on those things. Simply, what is nutrient use efficiency? Of course, there are a number of definitions. You come to the last one. So the simple agronomical definition is, what is the crop yield increment for use of unit amount of fertilizer? 1 kg of fertilizer if used, what is the kg of grain yield you are going to get additionally? In a simple term, in a farmer's term, what is the money you are going to get in terms of rupees for a rupee you are going to spend on fertilizer? That's called nutrient use efficiency. So finally, what is the additional rupees you are going to get by spending 1 rupee on fertilizer? So nutrient use efficiency at a crop level can be increased by increasing the available nutrient status in the soil, or increasing the nutrient uptake by plant, or even nutrient assimilation efficiency. If you increase any one of them, there is going to be increase in nutrient use efficiency. Humic substances are protein hydrolytes, microbial products, seaweed extract, and biopolymers, a number of biostimulants which we are listed, all of them work directly or indirectly in increasing the total nutrient available for crop uptake and the total nutrient taken up by the plant, and also influences nutrient use efficiency. So the nutrient availability is more, the uptake is more, and also nutrient use assimilation is more, resulting in more yield or more output for the unit amount of fertilizer with nutrient which is already there is improved by biostimulants, in addition to their own effect, as I told, a number of other role it has to play in increasing the plant physiological processes at cell level, molecular level, even at gene level. Let's take one by one how it influences nutrient availability in the rhizosphere. How biostimulants helps in increasing the nutrient availability. So nutrient fixation, I don't want to go into the detail because a lot of my people have already spoke about these things. Nutrient solubilization. For example, humic substances helps in nutrient solubilization. It holds the nutrients. It inhibits leaching of nutrients away from the rhizosphere, away from the root zone. It supplies the nutrients, whatever is available, so that it protects from leaching, and also it holds it and supplies for the uptake of plant. In addition to that, some of the, for example, seaweed extract, to take a bit protein hydrolysates, they also supply the nutrients in the organic form. So the plant can immediately take it up and use it for its biosynthesis of other macromolecules which are necessary for plant growth and development further. The next one is nutrient uptake. So biostimulant increases the total nutrient available in the in the rhizosphere. In addition to that, there are a number of ways by which biostimulant directly act on the nutrient uptake by increasing the root activity. Most of the biostimulants induces root growth. Humic acid is highly effective in inducing stimulation of root growth. It increases the root depth. It increases secondary and tertiary root formation, so that the total absorption surface area for water and nutrient uptake increases in the presence of humic acid. So many of these biostimulants also helps in that way, so that the total root available, total absorption surface area of nutrient and water increases in the presence of biostimulants. In addition to that, now there are a lot of information available to show that it acts at nutrient uptake. They're called as transporters. These nutrient transporters are present in the root. The transporters are called as carriers, channels, and also pumps. So these are the paths through which nutrient has to pass from the rhizosphere through the plasmalemma into the cell, so that it will carry it further into the shoot system. So most of the biostimulants helps in activity of these transporters at root level. So we can go on telling what, how, etc. A lot of information is now made available in the research in the recent past after 2015-16, how these biostimulants act at molecular and gene level to induce the nutrient transporters activity also. And for example, there are some novel proteins, transport proteins which are expressed by mycorrhiza, etc. So there are a number of ways by which it influences, stimulates the uptake of nutrient. So first, it increases the availability. Second, it increases the uptake of nutrients also. So nutrient assimilation rate. So nutrient is made available. So the nutrient which is already available in the soil, in addition to that, what we are going to put, and also what it is going to hold, and then supplying for the root growth, plant growth. In addition to that, it also helps in the uptake of nutrients, so that the physiology and biochemistry of the shoot system increases in addition to growth on the root. So it also increases the nutrient assimilation. the carbon and nitrogen metabolism of plant increases by the presence of biostimulants. For example, if you see at a shoot level, most of the times the pigment concentration increases, the chlorophyll pigment increases, the carotene pigment increases, there is increase in rubisco activity, there is increase in the nitrate metabolism, increase in the carbon metabolism, resulting in increase in growth development. The canopy increases, canopy size increases, the total canopy photosynthesis increases, resulting in higher growth and higher productivity. In addition to that, it also helps in translocation of these photosynthetes to economically important part. So at each level, these biostimulants are useful in increasing crop growth and development and finally productivity. So a lot of work has been done. What is the influence of biostimulants at molecular level? What happens at cell level, individual cell level? What happens in physiological? What happens to phenotypes? What happens to leaf area? What happens to branching? What happens to tillering? What is the amount of photosynthesis being translocated to the reproductive organs? What is the effect on grain size, grain number? So each one of these processes increase because of number of other effects of biostimulants in addition to its nutrient mobilization, nutrient uptake, and nutrient use efficiency at plant level and at canopy level. To give one example to end my talk, we have used, this company, really this work has been done by Balehonur Coffee Research Institute, ICR Institute, where unsolicited, we are not given any money for the test. They themselves tested the effect of one of these biostimulant humic substances on development and yield in coffee. You can see, so they used one of the biostimulants, humic substances, with only 75% of recommended dose of fertilizer or with 100% recommended dose of fertilizer with humic substances. You can see the yield. There is a significant yield increase in the presence of biostimulants, even though there is a 25% reduction in the recommended fertilizer dose, the yield is similar to that of 100% or little more than 100% of recommended dose of fertilizer. At 100% recommended dose of fertilizer, using a biostimulant like humic acid increases the yield further. In addition to this, it also increases the quality of the bean. Coffee bean, the A grade bean increases, so that it fetches more value again, and also increases the leaf nitrogen. With 100% recommended dose of fertilizer, if you also give a biostimulant like humic substances, there is increase in nitrogen uptake, clearly proving that it increases mobilization, it increases uptake, and also it increases the nitrogen use efficiency or nutrient use efficiency. So it reduces fertilizer inputs, biostimulants. It also increases nutrient use efficiency, the amount of yield you are going to get per unit amount of fertilizer used, in addition to if you use a biostimulant, and also it is one of the cost-effective input for an organic agriculture, and it is environmental friendly and reduces pollution. Thank you.
Good evening, everyone. At the outset, I would like to thank Team BIPA, especially Dr. K.R.K. Reddy, Dr. Raj Agarwal, Dr. John, for inviting me to give this presentation. So I will already, Dr. Annapurna has set the tone and made my job very easy. She has already informed about how these microbes work. You know, when we talk about microorganisms, I'm very astonished, as Dr. Annapurna was also saying, that why they have not been included. Dr. KRK was also saying in the morning that there are so many issues related to microorganisms. But one thing is very important is that, you know, in my first lecture in university, I say that plants and animals cannot survive without microbes, but microbes can survive without us. So that is the importance of microbes on this planet. So biogeochemical cycling and nutrient waste degradation, agri-waste degradation, that all Dr. Annapurna was talking about, it is such an important thing. That is, they create the vital juice for the plants in the soil. without their juice, that vital juice, these we call, they cannot survive, these plants in the soil. But I will focus my presentation only on abiotic stress and particularly on salinity. So, soil salinity, we are working on that, Gangetic Plain, one of the most fertile regions of our country, but it is suffering now from degraded lands, soil salinity. I think Gangetic Plain and Indus Valley, these two rivers provide food to maximum population in the world. But both these plains are in trouble now because of degradation of land, and soil salinity is one of the biggest issues. As you see, FAO projects that soil salinity is a very dynamic process and it is going to increase by about 50% by 2050. So already 7% of the land on earth is suffering from soil salinity, and that is equal to 20 times the area of France. I was in Uzbekistan just one day before. Half of the Uzbekistan is suffering from soil salinity. And in India also the situation is not very good in terms of soil salinity. 10% of the food security. If we talk about food security, the Sustainable Development Goal 2, and 10% is the reason behind food insecurity around the globe is soil salinity. So it is a very important aspect. We have to think over it. Dr. KRK is also already developing products, and there is one product on Hello. We are also working together to develop more such products especially for saline soils and degraded soils. So you see, almost every province in India has saline soils now. And I was talking about Gangetic Plain in Uttar Pradesh. So I will show you some pictures. See, this picture shows these are the original photographs of those Gangetic Plain in Uttar Pradesh around Lucknow and Kanpur region. Totally saline, marginal farmers, very poor, and almost zero productivity in terms, because if they grow, they are in loss. So they do not grow. They instead means it is better for them to be laborers in the cities rather than cultivate some crops. So we have a project from DST, funded by DST, where we have adopted these villages, around 20 villages in Uttar Pradesh, and providing them with some bioinoculants. We call them biostimulants. That is a long debate. I will discuss about that in just a few minutes. So we see, I think, so you see, we work on fluorescent pseudomonads, and we developed, I think these are fluorescent pseudomonads which we isolated from these saline soils. I think wrong PPT has been uploaded. They also show very good biocontrol. Now, fluorescent pseudomonads, what will we call them? As a biostimulant. We have isolated them from saline soils. They do direct activity, as Dr. Annapurna was saying. They show direct plant growth promotion by several activities like IEA production, by production of other metabolites, by several other activities. So, and some of them help in abiotic stress management for the plant. I will show you this one. I didn't want to show this PPT, although I think. Yes, some slides are missing from this PPT. This one I sent from, the original one is with, I don't think that has been uploaded. So, but I will manage from this. So these pseudomonads, what will we call them? Biostimulants, and now they are showing very good antagonistic activities as well. Extremely good antagonistic activities, I have checked. They are showing very good IEA production. They are showing very good siderophore production. They are showing very good, even some strains are showing very good ACCD activity. So if we check just one aspect, They are very good biostimulants. Then I checked them in our lab. Where is the PPT? So, yes, this one. So they are showing very good antagonistic activities also. So they are biocontrol agents as well. So there is a very thin line between what you will call biostimulants or biofertilizers or biopesticides. So that we have to think over it in a different manner, means how it is not that simple to categorize one strain as this one is going to be this, unless we do either we go for whole genome sequencing and check all the characters. Otherwise it becomes very dicey with some of the microbes showing diverse type of activities. So we work on these fields, you see. Barren soils, we developed some, we have meetings with farmers. This is the original picture with farmers. You see their soil here. So we generally go for in these villages, isolated these microbes. Already Dr. Annapurna has discussed about it, so I will not go in detail. So these can now be used for so many other things like biodegradation, bioremediation, as Dr. Annapurna was saying, agriculture waste degradation, and also for combating climate change, because carbon in this soil is very low. In these soils, carbon is very low. So we have to think about the climate smart agriculture with the climate change happening so quickly. This year, Dr. Annapurna is from Delhi, I am from Lucknow, North India. 40-degree temperature touched 40 degree in March. So wheat crop there was a huge decline in the wheat production. So we have to think over about the climate smart agriculture and how microbes can help in that. So we selected these strains. I will not go in the mechanisms and one more thing is that these areas are also suffering from pathogens. So there is a double attack, abiotic stress along with the biotic stress. So we check all these, as I was saying, showing very good, apart from being very good salt tolerant strains, you see, they can inhibit at very high salinity. So they can inhibit the pathogen also at very high salinity. So I will not go in the mechanisms, and we prepared some formulations using we train the farmers also. We do regular training with farmers so that they can produce some of these bioinoculants in their backyards and apply them in the fields. So we did experiments with different crops. I will just go through because of the time shortage. So almost from negligible, we have a very good production of chickpea. We did experiments on sunflower. All these are done in farmers' fields. You see, this is the situation of the field and nothing growing around. So we developed these inoculants using microbes. We also have bacillus strains with us, and we did experiments on wheat. Here is the treatment. You see, some farmers, they do not come immediately. They are not confident enough to come with us. So initially, some farmers joined the project. Then more are getting. So at present, the situation is for us, it is like, very good situation when all the farmers have not connected with us, because we get very contrasting pictures with adjacent fields lying white like this, low on carbon, low on nutrients, and the one which we have treated like that in the behind. So these are the drone pictures of those areas where we have done bioremediation using these bioinoculants, using microbes, production of wheat, improvement, and also some rhizobial strains. Professor Annapurna is an expert of rhizobia. We have got some novel strains from these very regions, highly saline soils. We developed some new methods also. And these are the exopolysaccharides Dr. KRK was talking about. They help in building the soil, means soil aggregation, soil biofilm formation around the roots, as Dr. Annapurna was saying about quorum sensing. So quorum sensing and quorum quenching both play very important roles in the field to attract the microbes. Now here I would like to add one more thing, that in these fields we checked 80% of the legumes were not showing nodulation. So that is the situation in these fields, which we call the fertile gangetic plains of our country. 80% of legumes not showing nodulation. That means either, as madam was saying, Either the rhizobial population is very low, or it is not able to form the nodulation because of the abiotic factor, abiotic stress, and lack of quorum sensing. Maybe we can work on that also. So all those things. So these are very interesting strains. We did experiments with non-legumes also using rhizobium in saline conditions. And you know, in saline conditions, you cannot use pesticides or fertilizers like urea or DAP like that. It will even aggravate the situation. So we can only go for biologicals in such soils, such agroecosystems. So that improves. All this work is published in several journals. So this helps in decreasing the EC, decreasing the pH of saline sodic soils, increasing the organic content. And this is how within 2 years, after repeated application and with different crops which we worked, now this has become quite fertile. Earlier it was not growing anything, the yield was extremely low, but now these farmers which have connected with us, they can grow vegetables also, salt sensitive vegetables even, like pea or tomato, like those. So these are some products which we have developed, although not commercialized, only for farmers through this project, DST funded project, which we are working on. So, but we have partnership with Shri Biostatics and a tripartite MOU with BioAgri. So maybe this will be commercialized very soon. So this is our journal, which is, I am editor-in-chief of Springer Nature. This is my team. We work with my research scholars and all, and regularly visit these villages. Thank you.
Good afternoon, everyone. I'm very glad to be here among the stalwarts of the industry, and I'm really feeling so thankful for giving me this opportunity by all the BIPA members, and selecting me means as I was just remembering like one small one Telugu proverb, like Hanumantudu mundaru kuppi gantulu. So like that, I'm too small in front of all of you. But yeah, I have been working with Dr. M.S. Rao, the expert in the industry of biopesticides, means who have been transferring the technology for many industries, and I have been seeing how a lot of BioAgri input industries have been started and growing. And initially I had a controversial feeling like whether these bio-inputs are really going to work, because whether all the industries are able to maintain that quality and all. And that was the time actually I have been discussing a lot with my guru, Dr. M.S. Rao, like how is it going to be, the future is going to be for the biologicals. Because there is everywhere, the farmers, where we go, we have to train the farmers, and we are really finding it difficult. In the beginning, it was like that, and slowly the situation has transformed, and a lot of government schemes and means the government support has come towards the organic agriculture. And seeing all that, I was more encouraged and to get into the BioAgri industry, whereas my basic research was on the cell-free culture, filtrates and all, mainly working on, because like we have been seeing a lot of restrictions for the microbes that they alone are not able to perform, because most of the times there are, it can be the shelf life constraints, or it can be the climatic conditions and all. So we were that was the time actually we were discussing like how we can overcome this kind of a situation, and that is the time actually we were thinking about, actually the microbes are wonderful organisms, but the thing is that these organisms are wonderful because of the miraculous molecules that are producing within themselves. So that they are which are actually it can be the enzymes or vitamins, or it can be any cell-free extracts, means many secondary metabolites are being produced by the microbes, which many of them which we don't even know, and that is why they are actually miraculous, we say. And what I have been seeing is like you all know that these microbes have interchanging roles, and whatever the molecules that they are producing is also having interchanging roles at different concentrations and at different stages of the plant and at of the microbe also. Like what the microbes does, microbes are actually, when they are induced into the plant rhizosphere, they actually induce some special mechanisms using some elicitors, which are actually plant signaling molecules. And because of these molecules, they are wonderful. They are actually creating wonders, like it starts maybe a nutrient uptake from by the plant, or it can be a plant defense mechanism, ISR, and systemic acquired resistance. And already ma'am has told, means most of the talk what I wanted to give the introduction, it was already given by the experts before me. And I have just given like how the microbes do work in the soil ecosystem. But when these microbes are induced into the soil, they have this mechanism, and why can't we take these molecules itself as elicitors, and we can actually make a formulation out of it. And by the time we started doing research, we have understood that there are many good researchers are already happening across the globe, and we started working on all of this, and we have understood actually these molecules will have a longer shelf life and will have more efficiency when they are used directly in the plant ecosystem. And coming to about how actually, that's what actually I wanted to say, that what is the difference between the single cell organisms and also the extracts. Actually, coming to the bioproduct, we know that single organisms are efficient, but when they are in consortium, they are even more efficient. So that is what we have seen. And more than consortium, we have also seen that these molecules, when they are used together, means these organisms, when they are grown together, they are producing certain useful hormones and enzymes for the plants, which are more efficient and are efficiently useful for the plant nutrient uptake and also the systemic acquired resistance. So we have, if we see that there are different categories of cell-free extracts, that are secondary metabolites, proteins, enzymes, vitamins, and lipopeptides. Like cell-free systems provide an alternative for the production of biostimulants. So these are categorized mainly into four broad ranges, that is actually for the growth promotion, biotic stress management, abiotic stress management, and quality improvement. So they are categorized based on the performance. It's like for the plant growth, auxins, gibberellins, etc. like, and organic acids and siderophores are having effective role in the growth promotion. And coming to the abiotic stress, the cytokinins, ABA, and ACC deaminase, these kind of molecules are having very efficient role in the abiotic stress management. Coming to the biotic stress management, that biosurfactants and secondary metabolites produced by these organisms like chitinases, proteases, and all are having very efficient role in the biotic stress management. And coming to the quality improvement, plant hormones like auxins and vitamins, antioxidants have a very good role. Actually, why we have to go for cell-free extracts compared to the organisms is because there is the physical barrier is not there. They can directly pass into the plant system. So that is one advantage, and eliminate the cell viability constraints, because that is the major constraint in our industry. Like we see the shelf life, because of the temperature and warehouse conditions, we are not able to maintain the consistency in results. And these molecules will have the consistency, and because they can work in very small quantities also, and provide direct access to the reaction conditions. And actually, if we talk about in the point of industrial usage, means for production, we can see that the complete reactor volume can be utilized for producing these organisms, and they can give a consistent quality results compared to the single organism formulations. And sustainable quality results can be achieved very efficiently using these molecules. And coming to the vitamins as biostimulants, So I have taken two examples like vitamins are mainly classified into fat soluble and water soluble. The fat soluble ones are mainly are having more antioxidant properties, and water soluble ones are having main enzymatic cofactors. And exogenous application of these has direct effect on the plant mechanisms. They can directly get into these means instead of the microbe getting into the soil ecosystem and then producing the elicitors, it directly works on the mechanism because of the cofactors and enzymes that are directly involved in it. And coming to the vitamin C, so it works against abiotic stresses like salt stress and also drought stress, and alpha tocopherol, which has direct effect in the yield increase. This is very prominently known among the industry, but if you see, due to their properties, vitamins are the excellent options for research, because I have seen very few of the molecules are being explored, but when we have gone for complete analysis of these molecules of the secondary metabolites, the crude extract of these microbes after the fermentation, we have seen that there are wonderful molecules that are coming out, which are very less explored. So that is one of the opportunity where we have and we can actually work on. And coming to the enzymes as biostimulants, my enzymes are mainly used by the plants at different stages, and they are involved in different mechanisms, like we can say starting from the nutrient uptake and production of the secondary metabolites for plant defense mechanism. But if we see that these mechanisms, we are actually targeting these enzymes which are coming to the last stage of the defense mechanism. Okay. So proteases, aminopeptidases and phosphatases, like phosphatases are actually mineralizing activity, and aminopeptidases are into the yield increasing activity. This is what we have been seeing. And cell-free extraction methods, mainly relating to the industry, we see the major water-based extractions are more efficient, and using different filtration methods which have more, which can yield phytohormones like activity, rich in phytohormone activities, and ultrasound assisted extraction, where we see that ultrasound waves at higher frequency are used and can produce phenolic compounds, and that is for the extraction of phenolic compounds, and enzymatic assisted extraction, which is used for the extraction of antioxidants present in the cells. And pressurized liquid extraction is another method where we go for protein extraction. So I am giving one of the examples, like using the substrate based production of these secondary metabolites. Penicillium binofilum, if we take as an example, using a different substrate, it produces different concentrations of lectins and also phenazyne and deaminase enzymes, which are actually useful in plant defense mechanisms against the pests. And coming to the microbial co-cultivation method, actually this is another cheapest and very useful method in the industry, where we can actually say that these enzymes can be produced without using any additional stress. We can just co-cultivate the microbes, like Trichoderma and Bacillus is just one example I have given, that when these microbes are grown together, they are like friends, means they produce very good enzymes and hormones which are more useful for the plants as well as for themselves. Whereas when they are grown individually, the effect of the same microbe we have found is not being used to its full potentiality. So these are some of the cell-free biostimulants that we have been seeing in a lot of literatures. So that is mainly the indole acetic acid and indolic compounds, mainly which are useful for the plant growth promotion. And coming to the plant protection, that we have been seeing a lot of siderophores and also surfactants and amylocyclines, which are mainly useful in induced systemic resistance. These are some of the simple experiments that we can do at our laboratory level itself, just to explore. Yeah, so just to explore whether the organism that we are using has any biostimulant effect or not. Just using them using the cell-free culture filtrates, we have, these are some of the tests that we have done. This is again is the nematodes and on the plant growth activity. Yeah. As I was given, I was asked to make 6 slides. I have made it only 6. No thank you slide. Thank you all.
Hi, very good evening to everybody. I should call BioAgri as knowledge का महाकुंभ. इतना knowledge मिल रहा है कि sorry I am speaking in Hindi, but so much of knowledge everywhere. I am just taking in. Last year was my first year at BioAgri, and this is second year. And since then, thanks to everybody at BIPA, Dr. KRK has introduced me to BIPA last year, and since then till today I have 10 products in microbes. So thank you so much. The volumes are built up. Okay, let me start my presentation. My presentation is all about regulations affecting biostimulants. Yep, that's the first slide. Thank you so much. And as the slide says, we are talking about whys and whos. So when we say whys, we're talking about why it was introduced. And we say whos, so who was lying behind it to get it introduced, and then who are getting affected because of those ill-licensing work. Sorry. Why? Because it was needed. Because a lot of spurious material was coming into the market, and it was affecting the farmers, affecting the trade also. So it was the need of the hour, and I thanks Ministry of Farmer Welfare that to bring that regulation into place. When this was introduced, probably at that time, INM was not clear what is the industry size, basically? Who were the stakeholders? Can you run the next slide so that, or I do it? Yeah. So I think I remember my meeting with Dr. S.K. Malhotra in the month of August 2021. And he asked me a simple question, Sahil, what is the size of your industry? So I told him that time, sir, there is no data as of now, but I foresee that there will be around 4,000 to 5,000 companies involved in producing biostimulants. And it is quite true, sir, because the way the G1s are coming for G3s, as of now, I think 35,000 G1 application has been received by INM, and they have successfully, though it's a very tedious process, and gradual issuance are there. So 300 companies, as per my update of, say around last week, 300 companies have been issued G3 successfully in India, and that's matter of applaud. Now, when we talk about the industry, this is the size of the industry. So we talk about cottage industry, 30% approximately, MSME, 60%, and 10% are large Indian companies or MNCs. MNCs also supplies to large Indian companies their products. So that's the category of their products. Now, when we talk about cottage and MSME industries, here the main issues are there because इनके पास पैसा नहीं है, they don't have money. They don't have money to go through talks. They don't have money to go through the other regulatory things which were imposed. Why? Because they want to stop spurious material laced with pesticides. So hence the pesticide residue was kept at 0.001% ppm and predominantly taken from Fasai. That is used for drinking water. Here we are using our product, diluting in the water and then spraying on the plant. So that need to be remodified or reworked on. I'm sure there are so many people around who can debate on that. And second thing is the talks. We want to stop heavy metals. So why talks? Can't we just go on label claim? Can't we just go on MSDS? There's a simpler way, at least for those products like humic, like seaweed, like fulvic acids, like your amines. And here, the organization called NBIF, Association, which Dr. Peter has already given a brief on it. I'll just start it with that. We have, as of now, 12 associations part of NBIF. It started with just 6, and BIPA was the first member of that. And we are of 12 associations, and we are having about 2,000 MSMEs as a part of NBIF. So coming back to the regulations part of it, it is not easy for even INM to understand, because a lot of gaps are there, a lot of misreadings are there in terms of industries, in terms of the products, because the categories they have introduced is primarily for the liquid part. They have not focused upon the granules, they have not focused upon the powders and other parts. So here the role of associations becomes very, very important. And so this is a slide basically I started talking to you earlier. So here the part becomes very, very important of association. And so changes are coming in INM. People are understanding the plight of cottage industries and MSME industries. They have soft down. And I would thank Dr. S.K. Malhotra in that, because he was a pioneer in meeting us. He was the first person to meet us, and from there the journey started. The current Joint Secretary, Mr. P. Ranjan, is very, very cordial for us, and he is understanding it. And many changes are coming into a place, because finally we were allowed to be part of CBC meeting, and we have put in our CBC is Central Biostimulant Committee, which has been formed like a CFC in fertilizers. And we have paced our request, and we have informed them that biostimulant is a very niche market. It is not as big as the pesticide market or as a fertilizer market. It's very niche. And to make those stringent norms for the industry to follow is very difficult. So why can't we go on certain softer norms? So they are working on it. They have recently formed a technical committee which is working on it. And we have given them a lot of documentary proofs in terms of humic, amino, seaweed, and fulvic acid, which are non-toxic. And they are working on it. We have given them also the testing methodologies on it, so they are working on it. So predominantly, things are shaping up. The role of BIPA is very, very important, because I would say in a word, BIPA is a technical backbone of NBI. A lot of inputs come from Dr. K.R.K. Reddy, Dr. Peter, and Mr. Srinivas. And so with their support, with their help, we are moving ahead on that. And the current Commissioner of Agriculture, and we had a opportunity to meet him last week, and he said very good thing. Now from ease of doing business, we are moving to live and let live. So Geo or Gene Do is a new buzzword of INM, and they have told us that we will not let MSME industry die because of the new stringent norms. Can I move to next slide? Yeah. So everybody is aware of that. G1, G2, and G3s. So shall I skip this slide or? Now, actually, we are on G3 because people have applied for G1 and they are getting G3s. But G3s, people are getting it, but don't be so happy with G3s because ultimately you have to go through G, that is registration of the product. G3s are basically a provisional registration, and one need to also register their product if they're selling other states. So it's again a commercial issue. So I request Dr. Saab to please help us in solving this issue also, so that we should not be registering it again in other states. Once we have got the G3 from INM, from a central fertilizer controller, it should be common for all the states. It's basically more of ease doing of business. Can I have the next slide? So these are the basic, this is the one. So these are the basic concerns that we all have faced. Now there's a rift coming because out of 20, this is the old numbers. Now after 35,000 applications, 30,000 means each brand is one application. So out of 35,000 applications, 300 companies have got the G3s. So now it's creating a rift between those who have G3s because they are marketing in a different way that, hey, we have a G3, so our product is very safe for humans, environment, and everything. And then those who don't have a G3 because there's a lot of pressures coming from the trade, lots of pressure coming from the state departments because they are disallowing those companies who don't have G3s. Second is that what we are saying is there are about 25,000 products in the market when talking of biostimulants. And we have just a handful of universities. So when we're talking of bioefficacy report, how can we generate that in a short time? It's next to impossible. Because when we approach a university, it says that they have other work to do, they have other work to follow. They don't have proper infra, they don't have proper support system to conduct all these things. So these are one of the concerns that we are facing. And hence we are suggesting them that let's go for control data instead of bioefficacy data. Let's go with pod control data. Or instead of trials at 3 different locations, go with 1 location trials. So this will help the industry to come on the track. Then the tox data. So we have been requesting them there should be no tox data for humic, fulvic, seaweed, amino, vitamins and other organic compound because they are being considered as a natural, been used since ancient ages. I'm missing Mr. Khurakiwala, otherwise he would have also supported me on that. Because these are the very natural compounds. So toxicity here doesn't play much role in that. And we are requesting ministry to help us with 4 or 5 products. And now here, the role of NBIF becomes very important because even at INM, there are having some issues in terms of, not issue I would call it a lack of manpower. So what we are doing it, we are matching the points. Like NBIF is meeting TIFAC. TIFAC have done about field trial reports on seaweed. They have conducted 170 field trials. So when we're talking about bioefficacy report on seaweed, so TIFAC already have it. So that report we have given to the INM. The CMCRI in Bhavnagar on the seaweed and NRDC on seaweed already have a patented product. So that product we are now through NBIF offering to INM so that they can study those products and those products can be included in the FCO. Similarly for potassium humate. So we are linking INM, we are linking ministry with different government-run organizations so that it can help them and us both. This is sum up the entire thing. So if anybody has some question on regulations, do we have time? No? Okay. All right. All right. Thank you so much. Thank you so much.